Right, so welcome to the class on microscopic organization of the histology. We are going to focus on the microscopic organization, the histology, as opposed to the whole of the digestive system. Maybe just to remind you of something that we've already covered in the gross lectures, but perhaps important to highlight, is that when you talk about the digestive system, we can look at the components in two ways. We have the histology, which is basically the alimentary canal. It is the root followed by what we eat from the oral cavity all the way to the anal canal. That means that we're talking about the mouth, the pharynx, the esophagus, the stomach, small intestine with its three parts, duodenum, jejunum, and ileum, then large intestine with its multiple parts, including the appendix, the cecum, the colon, the rectum, and the anus. So that is histology. The other part of the digestive system are the extrinsic organs of the digestive system, which focuses on the salivary glands, pancreas, liver, and the biliary system or gallbladder. This session will focus only on the microscopic organization of the histology. We will have another session where we're going to look at microscopic organization of the extrinsic organs of the digestive system. So again, a reminder about the structural organization of the gut wall. We've done this before, but I choose to repeat it here in this particular context. That we can look at the wall of the histology as consisting of four histological layers. Innermost layer is a mucosa. Up to that point. Second layer is a submucosa from there to there. Third layer is the muscular layer. You can call it Muscularis propria or Muscularis externa, extending from there to there. And lastly, we have the Adventitia layer, or you can call the serosa layer, the serosa. Let's talk about each of them. Starting with the innermost layer, the mucosal layer. The mucosal layer of the digestive system consists of three histological entities. We have the lining epithelium, basically the epithelium that lines the digestive system, therefore separating the lumen of the alimentary canal from its wall. Deep to the epithelium is the lamina propria. The lamina propria is a connective tissue layer just beneath the epithelium. And deep to the lamina propria is the muscularis mucosa. Muscularis mucosa is a thin layer of smooth muscles, a very difficult layer to sometimes pick, and so don't be surprised if you don't see them in the histology slides much. Very thin layer. We may also call this muscularis mucosa the muscularis interna. So those three are parts of the mucosa. Now, the lining epithelium in particular of the alimentary canal will either be stratified squamous epithelium or simple columnar epithelium, depending on where we are. The regions which are subjected to a lot of friction, like oral cavity, the pharyngeal region, pharynx, the esophagus, and the anal canal will have the stratified squamous epithelial type. you'll remember that particularly in the oral cavity, this stratified squamous epithelium may be parakeratinizing if you are talking about the regions of the oral cavity that come to contact with food. For example, the dorsum of the tongue comes to contact with food. The palate comes to contact with food. Some parts of the gum come to contact with the food. These regions will be lined by stratified squamous parakeratinizing epithelium, what we sometimes call the masticatory epithelium. Then most parts of these regions subjected to friction will be basically stratified squamous non-keratinizing epithelium, like what we talk about in the esophagus. Still some parts of the oral cavity, like the lower part of the tongue, the floor of the mouth, some parts of the cheeks will be non-keratinizing. The anus is also non-keratinizing. The rest of the histology, and this means from the stomach all the way to the rectum, they're lined by simple columnar epithelium. And so this becomes a very important distinguishing parameter to orient yourself in whether you are on the GAT or not. And yes, if you determine that you're on the GAT, then it can also help you to know which part of the GAT you're in. Of course, the choices could be wide. For example, if you note that it's simple columnar epithelium, then it doesn't speak much because it can be anywhere between the stomach to the rectum. But that's still an important distinction to make. At least you know it's not esophagus. At least you know that it's not the anus. So it's an important aspect, and I want you to be looking at the type of epithelium that is lining the gut wall as an important parameter in distinguishing histologically the parts of the gut. The other concept on this epithelium that is also important to note is that the epithelium of the gut wall have some scattered goblet cells. The number of goblet cells apparently increase as we move from the stomach all the way to the rectum. So what am I saying? You are unlikely to find goblet cells in the esophagus. You will find them in the stomach, but the number of goblet cells increase downward. And so this is an important thing to take into consideration. And you may have to train your eyes to learn to pick what are we referring to when we say these are lots of goblet cells and this is little amount of goblet cells. But generally, once you've seen a few slides, you will have a graphical memory of what a lot means and what little means. I particularly use this concept to distinguish between the large bowel and the small bowel. Large bowel will have a lot of goblet cells, and small bowel will have few goblet cells. It's an important parameter to use. Okay. Now, the mucosa has glands. The glands of the mucosa are tubular glands, basically. They are secretory portions being embedded within the lamina propria, like here. So if you look at this gland, its secretory portion is within the lamina propria, but its ducts opens into the lumen of the histology. And so it's possible to see these glands wherever they are found. A point to note again is that the GAT has mucosal glands. You can have a lot of them in the stomach. The ones in the stomach are particularly called the gastric glands. We also have a number of them within the intestines. The ones in the intestines are called the intestinal crypts of Lieberkühn. Take note that they open into the lumen. They may have a number of secretions depending on which site, which region of the alimentary canal you're talking about. For example, the ones in the stomach produce gastric juice. The ones of the intestines produce intestinal fluid or intestinal juice. Now, the mucosa of the GAT may have some characteristic folds which are unique to particular regions of the alimentary canal. For example, the stomach has rugae. Now, rugae are not primarily folds of the mucosa, but rather folds of the submucosa. But these folds are translated onto the mucosa. So histologically, how this appears is that you'll see a very thick fold of the submucosa. And because of that, the mucosa is also affected. You see an indentation of the mucosa as a result of an underlying fold. So the ones on the stomach are called the rugae. They are a bit irregular and in multiple directions. In some regions, the folds are longitudinal. What I mean is that they are vertical. They are along the length of the segment in question. In particular, there are some two regions which have longitudinal folds. The esophagus has longitudinal folds, and the rectum also has longitudinal folds. How is that important? We can use the presence and orientation of the folds to actually tell us which region of the GAT we're talking about. But you have to put into consideration how that section was obtained. For example, if you take a section of the esophagus longitudinally, then you are unlikely to see the fold because the folds are parallel to the section. So you are not going to see the fold. But if you take a section of the esophagus transversely, then you'll see the folds having been cut transversely. So understand the concept of how the folds come about and how they can be seen, in which sections they can be seen. So you'll not see the folds in all the sections. There are some particular folds that you cannot see in all the sections. Rugae, you can see them in all sections because they are irregular. Longitudinal folds are best seen on transverse sections, and circular folds are best seen on longitudinal sections of the gut. Again, some regions have circular folds. There are two types of circular folds. There are some circular folds that can be complete rings. We see this largely in the small bowel. They have different names. You can just call them the circular folds of the small intestines, or the valvulae conventis, or the plica circularis. They have different names, but they are complete circular rings. Some regions have circular folds which are not complete, and this is particular for the colon. So colon has circular folds, but they are incomplete circular folds. We call them hostrations. You are unlikely to see the hostrations on a microscope, so don't think much about it. The circular folds of the small bowel, you can see if the section has been obtained longitudinally. We have a few slides where that has been done, and so you can appreciate them. Now, these circular folds may also be of different amount depending on the segment. For example, for the small bowel, The jejunum has the highest amount of circular folds, and so we use that sometimes to distinguish between the jejunum and the ileum and the duodenum, for example. Villi are important characteristic folds of the small intestines. They are finger-like projections that increase the surface area for absorption. Whenever you see villi in the GIT, you know that you are in the small intestine. And if you don't see villi, start questioning whether you think that is actually small intestine. It's almost black and white. They're finger-like projections. So these are some of the things we can look at in the mucosa. One last thing on the mucosa is that the lamina propria of the GIT may contain regions where we have aggregation of lymphoid tissue. If such aggregations are not, well, usually they'll not be encapsulated. If their boundaries are not very distinct, then we call them diffuse lymphatic tissue. If their boundaries are distinct, then we call them lymphatic nodules. All in all, they all contribute or constitute what we call the mucosa-associated lymphatic tissue. So that can either be diffuse lymphatic tissue or lymphatic nodules, which are also called lymphatic follicles, lymphoid follicles. Now that we're talking about the gut, you may also call them the gut-associated lymphatic tissue. They may be present in the lamina propria. Some regions have a lot of them. Some regions have not so much of them. We see most of this in the pharyngeal region, particularly constituting the palatine tonsils and the lingual tonsils in the pharyngeal region. I have avoided to mention the pharyngeal tonsil and tubal tonsils because we are talking about alimentary canal in this context. So those regions have a lot of lymphatic nodules. We also have a lot of lymphatic nodules in the ileum itself and also in the vermiform appendix. Those become key things that we can use to actually pick out those segments of the gut, and especially for the appendix, the presence of mucosa-associated lymphatic tissue, and in particular, the presence of lymphatic nodules. Many of them help us to pick the appendix from the rest of the holoGT. If you see a lot of lymphatic nodules in a small intestine, it is most likely the ileum, because the ileum has a lot of them compared to the others. In particular, the ones of the ileum are known as the Peyer's patches. So that's the story of layer 1. Let's talk about layer 2. So layer 2, the submucosa, is a dense, irregular connective tissue layer, this one here, primarily responsible for the structural integrity of the gut wall. So the strength of the gut wall relies on the submucosa. This is because it contains collagen fibers, type 1 collagen fibers responsible for strength. Even from a clinical perspective, if a surgeon was to operate on the gut wall, when suturing back, they have to ensure that the submucosa is well opposed to ensure structural integrity of that region. The submucosa contains Meissner's plexus of nerves. The Meissner's plexus of nerves are a component of the enteric nervous system. found within the submucosa. So you can also call the submucosal nerve plexus. These plexus are responsible for the secretory functions of the digestive system. They may control exocrine secretions. They may also control endocrine secretions of the digestive system. Within the submucosa, we also have blood vessels, several of them, that provide vascularization to the gut. Now, something to note is that the submucosa of the alimentary canal generally does not have glands except on the duodenum and in the esophagus. So this becomes a very important distinguishing feature of these particular regions. When you see submucosa glands, when you see submucosal glands, then you know you are either in the esophagus or the duodenum if you are sure this is JT. Of course, there are other parts of the body, not necessarily limited to the JT, that may have some glands in the submucosa. So here we're talking about just JT. You may learn shortly that the histology of the esophagus can be very similar to some parts of the body, but the presence of the submucosal glands of the esophagus makes it characteristic. Those other regions may not have submucosal glands. Let's talk about layer 3. So layer 3 is the muscular layer. You may also call it Muscularis externa, or you may call it the Muscularis propria. Muscularis propria or Muscularis externa have smooth muscles within its wall, most parts having an inner circular orientation like here, inner circular layer of smooth muscles, and outer longitudinal layer of smooth muscles. Between those two lies the plexus of nerves that control contractions. This is the Albach nerve plexus. You can also call it the myenteric plexus of nerves. This is the second component of the enteric nervous system. Remember, the first one was the submucosal plexus. So the second one is the myenteric plexus, also known as Albach plexus. This one controls contractions. Some variations exist on the muscularis propria. For example, in the pharynx, the muscles are skeletal muscles as opposed to smooth muscles, although they are still involuntary skeletal muscles. The other variation in the pharynx, the fact that pharyngeal muscles will be more of inner longitudinal orientation and outer circular orientation. For the esophagus, the variation is that the upper third of the esophagus is largely skeletal musculature. The lower third of the esophagus is largely smooth muscle, and the middle third is a mixture of skeletal and smooth muscles. Even though they're skeletal, they're striated muscles, they're involuntary. For stomach, the variation is that instead of having only 2 layers of muscles, the stomach wall has 3 layers of muscles. These 3 will be named this way. We have inner oblique layer, the middle circular layer, and the outer longitudinal layer of muscles. This also makes the stomach wall to have a thicker wall compared to perhaps some parts of the alimentary canal. The unique variation in the colon is in how the outer longitudinal layer of muscles is organized, where this layer, this outer longitudinal layer exists in 3 bands. some 3 thickenings that we call the tinea coli. Lastly, the variation on the sphincters is largely based on the thickness of the inner circular layer. The inner circular layer becomes disproportionately thicker, much thicker out of proportion compared to the outer longitudinal layer. Those are present at sites of sphincters, like the cardiac sphincter, the pyloric sphincter, and the like. Layer 4. Okay, so the 4th layer is the adventitial layer, or the serosa layer. It's a thin layer of connective tissue on the outside. If it has peritoneal lining, then we call it serosa. If it doesn't have, then we call it adventitia. It's a bit difficult for you to see this layer on histological slides. And so I don't want you also to spend a lot of attention on that layer. Great. So having taken you through that, I want us to now look at the parameters that we'll actually check. We've already mentioned them, but I want to bring them into one consolidated slide to talk about the particular things that we check if you're going to identify a gut wall. We'll focus on mucosa, submucosa, and muscularis propria. So on the mucosa, what is it that we can check? Begin by looking at the type of epithelium present. And this is best appreciated at higher magnifications than lower magnifications. Look at the type of epithelium present. If it is simple columnar, then you know you're between stomach to the rectum. If it is stratified squamous, then you're either in the oral cavity, pharynx, esophagus, or anus. If it is not any of those two, you are not in the alimentary canal. The other thing we can check is the presence and the quantity of the goblet cells, and this helps us to distinguish largely between small bowel and large bowel. Large bowel having a lot of goblet cells and small bowel having fewer goblet cells. You can look at the characteristic because of false presence, and in particular, I want to emphasize on the villi. If you see villi, finger-like projections, you know you are in the small intestines. If you don't see villi, you are either not in the small intestine or you don't know how to see villi. So villi are characteristic features of the small bowel. The other faults, you may use them, not with a lot of emphasis much, like longitudinal faults of the esophagus and the rectum, you can use it, but I'll not put a lot of emphasis on that. The rugae are a bit difficult to grasp histologically because they are macroscopic faults, they are gross anatomical faults. So a bit difficult to grasp on histology, but if you see fine, I would want to imagine that if you see rugae, that's not the only thing you'll be relying on to identify that this is stomach. But if you see villi, that can be the only thing you are relying on to say this is the small intestine. So that's why I'm putting emphasis on some particular faults. The presence of microvilli is also an important thing to consider. Don't confuse villi and microvilli. Remember, villi are finger-like projections of the mucosa. Microvilli are still finger-like projections, but of the cell membrane of a single cell. How do microvilli look like histologically? At higher magnification, they'll appear like a railway track. And I'll show you a few. We describe that as brush border. So on the mucosa, I want to emphasize on those first four. But there are other things that we can still highlight on. The presence and amount of mucosal glands is also a thing to consider, especially if you have several mucosal glands. Start thinking about stomach, for example. Now, depending on how your cut is, you may also have a lot of glands in the intestines. So I'm not putting a lot of emphasis on the mucosal glands as a distinctive feature of a gut wall. The amount of lymphatic nodules is also a thing to consider. I'll put emphasis on this one if you want to distinguish the appendix and to some extent the ileum from other parts of the alimentary canal. In which case, this will be a lot. How about in the submucosa? Just one thing. The presence of the submucosal glands tells you you are in the esophagus or in the duodenum, if you are sure you're on the JT. For the muscularis propria, two things that I'll highlight, but I'll mention four. So the histological type of the muscles present is a key thing. If you see striated muscles and you know you're on the JT, then definitely you're either in the upper esophagus or in the pharynx. Or you're looking at some organs of the oral cavity. Because most parts of the alimentary canal will have smooth muscles. The volume of the muscular layer is a very difficult thing for you to use. We would imagine that we'll have thicker muscles at regions of sphincters and maybe stomach wall. That is very true. If you've not trained your eyes to pick what is a lot and what is little for muscles, you may be lost there. Also, the number of muscle layers is important. This becomes a distinctive feature for the stomach. If you are lucky to see the 3 layers of muscles, then you're sure you're on the gastric wall. The challenge is that it's very difficult to pick the 3 layers histologically because you're also not so sure how this section was obtained. Sometimes we take this section and so the circular and the oblique one almost both look oblique. They are obliquely cut, but in different profiles, difficult to pick on a slide. Sometimes a cut will appear that will capture all of them as oblique. And so very difficult to pick. I don't want you to put a lot of emphasis on that. But one last thing that you need to put emphasis on is look at the outer longitudinal layer of muscle and see whether you have bands or not. If you do have bands, then you are sure this is colon. And if you don't have bands, then you are sure this is not colon. Again, that is black and white. So having taken you through that theory part, I want us to now pick various segments of the alimentary canal. And I'll begin with this slide. I'll now want you to volunteer. So this slide, I'll give you 3 levels of magnification, but I want us to speak into each level of magnification. Let's start with this one. So the first question I'll ask you, which part of the alimentary canal do you think this could be and why? Okay, maybe I need to reverse the question. Do you think this is alimentary canal? And if you think so, then which part do you think it is? Okay, any volunteer, just put your hand up and I'll enable your mic. Yes, Michelle, you can unmute.
Is it possible for me to just make some observations but not say what I think it is?
Yeah, that's it, okay.
Okay, so I don't see any villi, so on this magnification anyway, so I don't think it's the small intestine. There's also no, I think they're called crypts of Leeuwenhoek, but I don't know if it's that, we just can't see it on this magnification. So I'm assuming that it's not the large intestines, and there's no mucosal, at least not that I can see, but I'm not sure if it's something you can see on this magnification. So I don't think it's the rectum. Yeah.
Okay, fair enough, Michelle. So I agree with you that it is not small intestine. It is not large intestine. I agree with you on that. Yes, Shannon.
Okay, I don't really have options, but it is look like inner lamellae. So I am going to guess that it's an artery and not GAT.
Very well. And you've picked it correctly. This is not alimentary canal. And I can give you a high magnification of that. So we see an internal elastic lamina present. Even a much higher magnification, you can see the internal elastic lamina present. The presence of an internal elastic lamina tells you definitely this is not alimentary canal. So yes, it's a luminal organ, but it is not alimentary canal. And as Shana said, this is particularly a muscular artery. Muscular artery has prominent internal elastic lamina, has a thick muscular tunica media, and has Adventitia. And you can see that from this. So this is the tunica media, that's the Adventitia. Very well, thank you, the two of you, for that. Okay, another slide. So this is a transverse section of a gut wall. Now that one, believe me, it's a gut wall, transverse section. This is the lowest magnification. I'll give you another magnification. That the second magnification. So the lumen is this side. You move like that. So that is mucosa. Then we are now in the submucosal layer there. Another magnification. So this one captures for you the submucosa up to there and the muscularis externa there. Okay. So you've seen the 3 levels of magnification. I'll return it back to this. So this is the mucosal layer up to somewhere there. Then submucosa up to somewhere there. Then we have the muscularis propria up to there. Then this is Adventitia. Which segments of the GIT could this be? And why? Anyone? Yes, we'll start with Faith.
Okay, I'd say the esophagus, because there's presence of the submucosal glands, and then the epithelium is stratified squamous, non-keratinized.
Very well.
Yeah.
Very well. And I'll actually just skip to those two. That from here to there is the mucosa. At this magnification, we confirm that the epithelium is stratified squamous non-keratinizing epithelium. So that rules out most parts of the alimentary canal. You are remaining only with oral structures, pharyngeal structures, esophagus, and the anus. Then, boom, we can see glands in the submucosa. These are the glands of the submucosa. The presence of glands in the submucosa tells you this is now esophagus, nothing else. You can also see some blood vessels, but these are common to all submucosal regions. They have blood vessels. So if you've not learned to train your eyes on how to pick submucosal glands, or basically glands, that's how they look like in the submucosa. This is the muscularis propria. So it's a transverse section. So that means that this is the inner circular layer. So if it's a transverse section, then the inner circular layer will be cut longitudinally, in long profile, basically. And then the outer longitudinal layer will be cut transversely. And that's what you see there. So yes, that is the slide of the esophagus. Okay. Now, this is still esophagus. Now the whole profile. If you look at the lumen of the esophagus, you'll see this fold, that fold, that fold, that fold. Considering that this has been cut transversely, it means that these folds were running vertically. So these are the longitudinal folds of the esophagus. I confirm to you esophagus because you can see the epithelial lining is stratified squamous, non-keratinizing epithelium there. And at this magnification, you can see the submucosal glands again present. This also still shows you the esophagus, the epithelial lining characteristic, and the submucosal glands. And that is still esophagus, the same slide. Now, most slides of the esophagus would be cross-section as opposed to longitudinal. So train your eyes on that. Great. Next slide. So we have a slide projected here. Question: which part is this one and why? Anyone? Yes, Antoinette?
I think this is the trachea
Okay, why do you think so?
because of presence of a ring of cartilage, like the cartilage.
Very well. Very well. So we can see C-shaped cartilage present there. And that C-shaped cartilage is characteristic of that of the trachea. I brought this up so that I tell you that most of the slides that we have of the esophagus will be slides that have been captured together with trachea. So when you look at them in the microscope, you need to know which side is actually the esophagus and which side is the trachea. This one is trachea and this is esophagus. This, however, has been displaced in this slide. Usually, where that C is defective is where the esophagus would be. So we expect the esophagus to be somewhere here, as opposed to where it is now. This has been translated. So usually the esophagus will be next to the trachea, adjacent to the cartilage defect of the trachea. Maybe this one would show you a better view. So this would be the esophagus and this the trachea. Usually adjacent to the muscle trachealis. So the slide that you'll be having more will be the slide of trachea and esophagus. If you're studying esophagus, make sure you are actually looking at the esophagus and not the trachea. All right. This is a slide of the gastric wall, stomach wall. And what do we see? So from here to there is the mucosa of the stomach. Then from here to there is a submucosa. From there, going that way, is the Muscularis propria. It's very difficult for you to pick this at the stomach at this magnification. I have the advantage of having acquired the slide and also the advantage of having seen many. But some things to highlight. One, we can see that the muscular layer is a bit thick compared to what we are going to see next in other slides. So it has a thick muscular wall. We can also note that the submucosa does not have glands. That's a bit general, but we've noted. Maybe you can see something there, that's a lymphatic nodule or diffuse lymphatic tissue, so we can just say it's part of malt. Again, not telling us much because we've just seen one or maybe two. There's another one there. I wouldn't call that a lot, and so it's not telling us much. At a higher magnification, we can see the malt there. Maybe there's another one there, and there's another one there. Importantly, from here to there is the mucosa. You can see some depressions on the surface. And you might be tempted to call this villi. They are not. And let me tell you why. A villi will have a broader base and an apex that is tapering, so becoming much sharper. It's like almost pyramidal. Like villi would be like this one, that single one. And I'm not saying these are villi, villas. I'm just saying that most of them will look like that. A sharper apex and a broader base. The ones we're seeing here are predominantly not of that nature. So what are these? The emphasis is more on the gaps between those structures. And those gaps are the ones we call the gastric pits, so they're present here. And then you can see some lines going deep. Those are the ducts of the gastric glands. And these ones are the gastric glands. So there are several glands in the mucosa. These are the glands. These are the ducts of the glands, and they open into the gastric pits. Submucosa is very thick, doesn't have glands. The Muscularis propria is thick with muscle. This is still a higher magnification so that you see the gastric pits. So these are the gastric pits. Now you can appreciate what I meant by these are not villi. You see, a villi will not have that kind of a broader apex on top. So these are not villi, but the emphasis on the gaps there, those are the gastric pits. So that is still stomach wall, and I want you to train your eye on seeing the gastric mucosa. All right. Occasionally, Now, going back to the slide, still we started on the stomach, you can see this fold of the submucosa. And because of this fold, there's a translated fold on the mucosa. So these are the ones that give us the impression of the rugae. Now there's that one, there's this one here, there's another one there. So because of this submucosal folds, the mucosa has those indentations, which we call the rugae. You may or may not see them on slides. Still another example of where the submucosa had a very thick fold of rugae, and so you have a large fold on the mucosa of the stomach there. Okay. I want us to discuss the gastric glands. This image still shows you the gastric glands, several glands. Here, the orientation does not allow us to see much of the pits. There are just few, but we see a lot of glands present in the mucosa. So these are all gastric glands. High magnification shows you that, yes, they're lined by simple columnar, simple cuboid epithelia. Their ducts are simple columnar. They empty into the surface. So let's talk about the gastric glands, or the gastric mucosa. We had talked about the cell types in the gastric mucosa. So we have mucous neck cells, which produce mucus. We have chief cells that produce pepsinogen. We have parato cells that produce acid, intrinsic factor, and gastropherin. The enterochromaffin cells produce histamine. The enteroendocrine cells produce hormonal substances, and there are different types. The G cells produce gastrin, the D cells produce somatostatin. Let's look at these secretions from their functional perspective. Most of those secretions will be part of gastric juice. Pepsin is obtained from pepsinogen. Once pepsinogen has been secreted, it's converted to pepsin. And the cell that produces pepsinogen is the chief cell. The role of pepsin is to break down proteins in the process of digestion. The acid of the stomach is in form of hydrochloric acid. It's produced by the parato cells. The role of the acid is to activate pepsinogen to pepsin, but it also has antimicrobial properties. Intrinsic factor and gastropherin are also produced by the parato cells. The intrinsic factor promotes absorption of B12 in the ileum. The gastropherin promotes absorption of iron also in the ileum. Histamine, which comes from the enterochromaffin cells, help promoting acid production via its H2 receptors rather than H1. The mucus present produced by the mucus cells, they could be the mucus neck cells, or they could be the mucus surface cells. Mucus cells produce mucus. These are the goblet cells that help in lubrication so that we prevent against corrosion. Great. Let's move on. So next slide. The question, what parts of the GIT could this be? Anyone? I'm waiting for a volunteer.
Take an attempt from what I see.
Yes, you can.
All right, so definitely we can see that it's a luminal organ.
Mhm.
And if I look at the lining epithelium in the lining epithelium, I think it's simple columnar.
Okay.
All right, when I look at the submucosal layer, I am not really seeing any glands, which means it can't be the duodenum or the esophagus.
Okay.
I'm also not seeing any villi, so it can't be the small intestine.
Okay.
Okay, I think I don't know. I'm seeing are those. No. I think I can't see that in that magnification. But yeah, I think that's what I've seen.
Okay. So where is your mind taking you with all this description? If you had to just say one thing.
Okay. So because I said that it is simple columnar, okay, that would mean that it has to be between the stomach and the rectum, and between the stomach and the rectum. And so we said it can't be the small intestine. And I'm not seeing we said there's the tinea coli, which would be present if it were in the colon. So it's not the colon either. And the outer membrane, it's not looking too thick. So I'm not really thinking of the stomach either. So I don't know, maybe that leaves the rectum.
Okay, so let's go with the fact that you don't think it's the small intestine, you don't think it's the stomach, you don't think it's the colon. Let's go with that one. Let me hear from someone else. Yes, Brian.
Okay. Mine might be a bit different because I honestly don't think this is actually in the GIT. And the reason I say that is because if you look at the epithelium, I will actually say the epithelium is stratified, and at the top, at the apical surface, you can actually see some cells that appear to be very thickened in some sort of way. So in my opinion, I think this might be probably translational epithelium, transitional epithelium, and therefore I would say this is probably the ureter.
Okay, fair enough, Brian. Thank you. So two things. So we're seeing a luminal organ, and the lumen here is that region surrounded by an epithelium that is not characteristic of the alimentary canal. That one I agree with you, Brian. That this epithelium is not simple columnar. It is actually stratified. It could be stratified cuboidal, stratified columnar, but at least not simple columnar. Maybe I give you people a higher magnification of this epithelium. And that is how it looks like at higher magnification. So can somebody identify for me this epithelial type and give me a reason why that's the identification? Anyone to identify the epithelium here? Yes, Blessing?
I think it's transitional
Okay, why do you think so?
because at the surface there's binucleated. Yeah, you can see binucleus.
Okay, so yes, there's some binucleate cells on the surface. And what Brian said earlier is also still true. The cells on the surface look like they occupy a large zone, a large surface area. So they're big cells. So this is transitional epithelium. In the context of it being transitional epithelium with this kind of a lumen, almost star-shaped, this is definitely ureter, as Brian mentioned. So this is not alimentary canal. Again, and we've seen why it cannot be alimentary canal. Okay. Next slide. So I want somebody to tell me one characteristic feature of this slide, and because of that characteristic feature, what it could be. Yes, Mark. Mark, you'll have to admit, yes.
Yes, so one characteristic feature of this slide is I can see villi, so which means that this could be the small intestine.
Very good. And that's what I wanted to pick. We've seen villi. This is small intestine. Okay. Another question, anybody? What do you think this could be? Yes, Michelle? You can unmute.
Does that just show that it's intraperitoneal? Is that the peritoneum?
Yes, you're right. So this tells you that this organ is intraperitoneal. That specific structure is actually the attachment of the mesentery. So this is where the mesentery is attached, and usually at the attachment of the mesentery, you see some blood vessels there. So you're right, this is an intraperitoneal organ. And that also perhaps tells us that most likely this is not duodenum. Because you know that duodenum is retroperitoneal predominantly. Well, we'll use other parameters to pick that, but let's say something about the mucosa of the small intestines in great detail before we look at the small intestines. So the mucosa of the small intestine is lined by simple columnar epithelium, just like the rest, most of the parts of the JT. There are different cell types on that epithelium, and these cells have different functions. For example, we have the enterocytes. The enterocytes are the majority of the cells that line the epithelium of the small intestines. What is unique about them is that enterocytes have a brush border, and I'll show you brush border shortly. Brush border is where we have now the microvilli, like these ones. So because of the presence of the brush border, we know that these cells are responsible for absorption of nutrients. They're the majority of the cell types. Second, okay, so this still shows you the enterocytes. A hemocreation, those are the enterocytes. They have microvilli. If you see these are villus, and that's another villus, so these are villi, the gap between them usually lead to a gland, which we call the crypts of Lebercon. I'll talk about the crypts shortly. In the previous image, this is a villus, that's another villus, and this is the crypt of Lebercon. So I want us to talk about the cell types on the villi first, before we talk about the cell types and the crypts. We have the goblet cells. Goblet cells produce mucus that help to protect against friction. Will also protect against toxins. Will protect against infection because the mucus picks them, hold them. So it protects not just against friction only, but even toxins and infections. So they'll be present, these goblet cells will be present at the villus epithelium. They're also present at the cryptal epithelium. As you can see, these are still goblet cells. We have the microfold cells. The microfold cells are found where the epithelium covers a region having a mucosa-associated lymphatic tissue. These cells function as the antigen processing and presenting cells. So like here, we have microfold cells on top here because down here, we have a Peyer's patch. So the microfold cells are basically the macrophages of the small intestines. Then we can have the intraepithelial lymphocytes, which are basically migratory lymphocytes from the surrounding mucosa-associated lymphatic tissue that is responsible for fighting infections. Let's talk about the cell types in the crypts. The intestinal crypts of Lieberkühn are tubular glands which open adjacent to the bases of villi. So between two villi, there's a gap there that leads to a crypt. There are several cell types within the cryptal epithelium. The enterocytes are still there. Now see the main difference between the enterocytes of the cryptal epithelium and the enterocytes of the villus epithelium in terms of function. The enterocytes here at the cryptal epithelium produce secretions. So it's a fluid that is being produced like that from this side to that side. Then that fluid comes this way. That fluid helps in absorption of nutrients. So when that fluid reaches here, the enterocytes of the villus epithelium now absorb that fluid together with the nutrients. Now, what do you notice? The role of this one is to take that fluid that way, and the role of this one is to take the fluid that way. So there's a circulation of fluid here. As the fluid circulates like that, nutrients are absorbed. It's a very important mechanism of nutrient absorption. Think about that again in the context of someone's villi have been destroyed. What will happen? The cryptal enterocytes will produce the fluid, but the villus enterocytes cannot absorb the fluid. What will happen? You'll have diarrhea. That's why anything that destroys the villi will present with diarrhea. And we are losing fluid in two ways here. The water that you swallowed from the mouth will not be absorbed, so you lose that one. But again, the fluid from the body released by the cryptal enterocytes will also be lost. And that is why this patient will present with dehydration. Well, much fluid will still be absorbed in the large bowel, in the colon. The colon still absorbs water, that's fine. So hopefully if the colon, if it's a lot, then somebody will get diarrhea, unless the colon is able to absorb as much. The mucus cells in the cryptal epithelium are similar to function like those of the villus epithelium. We have Paneth cells of the cryptal epithelium. These ones produce some substances which have antibacterial properties, so therefore immune functions as well. We have the enteroendocrine cells, which we can also call neuroendocrine cells. The neuroendocrine cells of the JT produce chemical substances which function as hormones. Examples of hormones produced by such cells are the gastrin, cholecystokinin, and secretin. I'll have a slide on cell types of the gut epithelium that produce hormones and a list of some hormones, so we can hold that story at that point. We have stem cells. Stem cells give rise to the other cell types. We may also have the tuft cells, as you can see in that list there. Now, tuft cells would be scattered. They could be at the villus epithelium or at the cryptal epithelium. The tuft cells function as chemoreceptor cells. They are similar to the brush cells of the respiratory tree. They function as chemoreceptor cells. They produce some chemicals, interleukins to be particular, that help in immune functions as well. All right, so those are the cell types of the cryptal epithelium and cell types of the villus epithelium of the small intestines. With that, we can look at different slides of the alimentary canal. So we start with this one. We see a luminal organ. This magnification is not very optimal to say much, apart from the fact that we can see probably a muscular wall there, and something that we can't pick much at this magnification. But at this magnification, we're able to pick something. So we see this is a section of the wall. The lumen is this side, and this side is the outside. This is a segment of the small intestine. I want a volunteer to tell me, give me 2 reasons why this is actually duodenum. Yes, Steve.
One reason is there's the presence of submucosal glands and the presence of villi.
Okay, so I agree with you on that. So remember, I already said that it's small intestine. So I'm looking for a reason to distinguish a part of a small intestine. So the question here is, why is it duodenum and not jejunum and not ileum? And so I'll take the first part of your response, that we can see submucosal glands. That one is true. So this is duodenum because of that. The second response just tells us that it's a small intestine, doesn't distinguish duodenum, jejunum, and ileum. So I'm looking for a second reason. I'll give this to Robert.
Because it's retroperitoneal.
Okay, and I want you to elaborate on what you mean by that from this image.
When we look at the outermost layer, we can see the serosa. It's just Adventitia.
Great. So we can't see epithelial lining on this side, so we can't see serosa basically. And so because of that, we can conclude that doesn't have, it's not intraperitoneal organ. Good. Another question to, okay, so I can see more hands up. I'm not so sure whether you want to respond to the previous one or fresh questions. Let me start with the, yes, Becky. New question, am I previous?
It was still the previous question on this slide. I just wanted to mention the presence of goblet cells in the mucosa.
Very well. And actually, that was my question. So goblet cells is present here, not helping us to distinguish whether it's duodenum, jejunum, or ileum, but it's an important observation. We'll expect duodenum to have goblet cells to prevent the against corrosive effects of the acid from the stomach. And so they are actually present on this particular epithelium. So those dots that you see there are actually goblet cells. Yes, Michelle.
If this is a longitudinal section?
No, it's a cross section. Remember,
Okay.
it was this one. So I just took this segment only.
Okay, so it wouldn't be fair to make the observation about any folds in the submucosa in this one because it's not longitudinal.
Yes, it will not be objective.
Okay.
Shannon?
I'm just wondering if I'm not wrong, maybe I'm wrong. You had mentioned that it is not usually that easy to identify the serosa, presence of the peritoneum. So I'm wondering, would it be okay to continue trying to identify part of the JT, whether we have the peritoneum or not?
So I agree with you that I don't want you to belabor much on looking for the Adventitia and not looking for it. I agree with you on that one. Except for this particular slide, I wanted to know whether you guys have that in mind, that yes, duodenum is retroperitoneal, and I'm putting this compared to the other slide where I actually showed you the mesentery. So the other one had a mesentery, so it's intraperitoneal. This one, we don't see any evidence of mesentery. We don't see any evidence of the serosa, which would be epithelial general, because remember, it's pavement epithelium, simple squamous epithelium that would be expecting to see there. We're not seeing anything on that end. So I just wanted us to pick that. I will not, however, use that as my first distinguishing feature of duodenum. So if I was to ask you why this is duodenum, I am satisfied with the answer of yes, it's small bowel because we can see villi and we can see some mucosal glands, and that is adequate. Except I brought that up for the sake of this complete learning in this session. Right, I'll give you another part. Okay, so this is still higher magnification of that duodenum, sorry. In this slide, we confirm, we can see the villi, we can see the goblet cells, and we can see submucosal glands. So this is the beginning of the submucosa. We can see several submucosal glands. So that is duodenum. Okay, we try another one. So let me give you a background. This is a longitudinal section of the small bowel. Why do I say so? Because I can see villi. Again, these are villi, several of them. But it's a longitudinal section, that's what I want to remember. This is a lower magnification, and that's a higher magnification. So you can still see the villi. That's one villus, that's another villus. It's a unique stain where the blue is representing collagen, connective tissue. Okay, sorry. Okay, it has already shown. But maybe the question is, why do you think this is jejunum and not duodenum? Okay, again.
Okay, I'd say it's jejunum and not duodenum because of the presence of the circular folds, the plexi circularis. Yeah,
Very
and
good.
not duodenum.
Yes, so we can see several of them. Well, remember, even duodenum will have them, except they'll not be this close. So these are very close circular folds. We hardly see a big gap between one fold and another. Like here, we would have expected a large gap there and a large gap there. We're not seeing that. So there are folds of the submucosa translated onto the mucosa. That's very key. Several plica circularis. The intimate nature of the plica circularis tells us that this is most likely jejunum. Again, it is not duodenum because we can't see submucosal glands. Okay. So this magnification still confirms that there are folds of the submucosa which are translated onto the mucosa. We don't have the appropriate staining, but that looks like a diffuse lymphatic tissue, that region there. And even that one. And probably that one. But this is not the appropriate staining protocol for lymphatic tissue. Yes, Sydney.
I have a question. Can you quantify this, the jejunum, if you use now the opposite for duodenum, the absence of submucosal glands?
Sorry,
Can you quantify this, the jejunum, if you say like that the absence of the submucosal glands will make it to be the jejunum and not the duodenum?
So the question was a bit specific. The question it asked was, why is this jejunum and not duodenum? So that's why the concept of the glands had to come in as well. Unless I'm not understanding your question, we can say this is jejunum because we can see the plica circularis are closely packed. And this is an assumption that we've already agreed that we are looking at the small intestine based on the presence of the villi. So the question is, could it be duodenum? Could it be jejunum? Could it be ileum? And we are settling on jejunum because of the intimacy of the plica circularis. And we are saying, yes, it cannot be duodenum because it does not have the submucosal glands. It cannot be ileum because of the intimacy of the plica circularis. These ones are very close.
Okay, thank you.
All right. Okay, so that's jejunum. Let's try another one. Any volunteer for this? What part of the alimentary canal could this be and why? Yes, Steve?
Just the child, I would say stomach, because I can see it has a very thick muscular layer. Yeah.
Okay, so you've observed a very thick muscular layer and you think it's stomach, okay. I saw Lucky's hand up. Lucky, what do you think?
Okay, mine was a bit different. I thought of a sphincter, but I don't have a specific sphincter in mind because of the thick muscular layer. Because, okay,
Oh, okay, so you think the sphincteric region, again, because of the thick muscular layer. And I love the fact that both of you have picked the thick muscular layer. That's okay. I want to hear from someone else. Yes, Sydney.
Okay, according to Steve and Lucky, they have 3 thick muscular layer, but I can see like there are 3 layers within the muscular layer. So I might assume it has, like, let's say oblique, inner muscles, then middle circular muscles, and outer longitudinal.
Very well. So we are adding to the fact that, in addition to the fact that it's a thick muscular layer, we are adding to the fact that, yes, we can see from there to there, it's one orientation of muscle. Then from there to there is another muscular orientation. And from there to there is another muscular orientation. So we have 3 muscular orientations. So we can use from there to there, 1, then this is 2, this is 3. 3 muscular orientations. So that will eliminate the possibility of it being a sphincter. Because sphincter will only have 2, and the inner one will be much thicker. We can still, based on that, entertain the thought of stomach, but at least we've eliminated the sphincteric components. Yes, Faith.
Oh, I wanted to say, okay, just hear me out, yeah?
Mm-hmm.
So then I think it's, okay, let me just start with why. So you see, it's a bit far-fetched, but like inside the lumen, I'm seeing like a brush border by mistake. So then I'll say those are like microvilli. Okay, and I can see the 3 circular folds and stuff like that. So is it right? Okay, I know it's from the male genitalia. So then I'll say, if we say the microvilli, okay, the brush border is like stereocele instead. That's crazy, but like I'll say the vas deferens. Thank you.
Okay, so Faith, I agree with you that this is not alimentary canal, except the reason you've given me is not very satisfactory, because at this magnification, you cannot see microvilli. Microvillus is seen when you have a high magnification of a single cell. Then you see microvillus. So you cannot see microvilli, neither can you see stereocilia at this magnification. But yes, I agree, this is unlikely to be alimentary canal, and let me just tell you why. If you look at the size of that lumen vis-a-vis the thickness of this wall, this cannot be stomach. Stomach has a very wide lumen compared to its wall thickness. So this definitely cannot be stomach. We expect the lumen of the stomach to be much bigger than its wall. This wall is much thicker than the lumen. Yes, Robert.
Okay, now I'm trying to like put it all together. Now that you say that the lumen is not that big, and then you have muscular layers, what I'm thinking about is either the ectocervix or the vagina. Yeah, thank you.
Okay, so the correct identification is actually what Faith talked about, what Agan talked about. I just didn't buy the reason she gave me. This is not vagina because, again, vagina doesn't have those 3 muscular layers. And again, vaginal lumen cannot be this size. The epithelium of the vagina is stratified squamous non-keratinizing epithelium. For cervix, again, the number of muscular layers is the one that discredits it. So the presence of 3 muscular layers, and the reason I put this is so that we can grasp the idea of picking muscular orientation. So these 3 are very characteristic for the ductus differential, the vas deferens of the male reproductive tract. Okay, we'll talk about that at another time. The aim was not to identify it as male reproductive tract, but the aim was to pick that this is not alimentary canal. That was the purpose of the slide. Yes, Martins. Oh, Martins, you muted yourself again. Just unmute again, Martins.
Okay, sorry. My question was, can you identify the epithelium from this what's it called, magnification, really?
Not quite.
Oh,
That's I'm
okay,
not
okay.
talking much about the epithelium, and that's why I was not very accommodative of the reason Agan gave me, because
Okay,
that reason relied on you seeing the epithelium properly.
okay. And the connective tissue around is that like, okay, that's connective tissue on the border of it, the edges.
Yes. So this is the surrounding connective tissue.
Okay.
The vast difference is located within what we call the spermatic cord. So spermatic cord has multiple tissues, and those are the other parts of the tissue, the spermatic cord. Okay, yes, Michelle.
You had also mentioned that a reason why this was not the stomach was the ratio of the lumen and the muscle. Could you please repeat that statement?
All right, so the stomach, we know, will definitely have a very wide lumen compared to the thickness of its wall. I'm looking at this and look at that wall thickness. Compare that to the size of the lumen. It's not very characteristic of what we know the stomach to be. And that's why I'm saying, even though this one has 3 muscular layers on its muscularis propria, it cannot be stomach wall. Okay. So we go to next slide. So this is a slide of the small intestines. This is low magnification. It's a longitudinal section. We see plica circularis there, another one there, another one here, and another one there. It's a longitudinal section, as I've already told you. We can see the villi. This is the submucosa, and we cannot see glands. So we know it is not duodenum. Based on the gap between this and that, the plica circularis are widely spaced. We can conclude that this is ileum. This is a hypertension of ileum, and we can see mucosa-associated lymphatic tissue present. We can see villi, and I want to note that the villi have a lot of goblet cells now. This magnification is important for us to single out the villus. Simple columnar epithelium lining the villus. This is goblet cell, that's goblet cell, that's goblet cell. So there are several here. I want you to pick that railway track appearance on the surface of the villus, and that's the brush border, telling us that this is microvilli. So the enterocytes have microvilli. We can see another one there. The enterocytes have microvilli on the wall, on the surface. All right, so this is the ileum, basically. I think I've already shown you that part. Okay, seems like a repeated slide. This is still small intestine, and particularly still ileum, a longitudinal section. The aim is to see the gap between one plica circularis and the next one. There's a big gap. We can see villi, yes. We can see plica circularis, yes. This is the muscular layer. We cannot see the glands in the submucosa, so we agree it's not duodenum. These are the villi. You can appreciate that they have broader base and tapering apex. The grooves between them constitute the crypts of Lieberkühn. Higher magnification display the brush border, which is characteristic of the presence of the microvilli. Okay. Yes, Lucky, you have a question. Let me enable your mic. Yes, you can unmute, Lucky.
Okay, I have a question. For the small bowels, we use the Peyer's patches to pick out the ileum. So is it that the jejunum doesn't have the lymphatic nodules, or they're present but minimal compared to the ileum?
So ileum has much more lymphatic nodules compared to the rest of the small bowel. And similar to appendix, we are going to see it also has several Peyer's patches, or rather lymphatic nodules, compared to the rest of the large bowel. Yes, Victor. Victor, you have a question? Okay, you are muted. Yeah, Lucky, you still have another question? Is it clear, the previous one?
Kidogo, yes. Kidogo. Okay, so by several, okay, how many are like a lot and how many are few?
Okay,
Because for the slides,
I get your point. So it's a relative thing. We cannot put a number. It's a relative thing. So this is something that's better when we have other parts, then we are comparing, rather than we just have one absolute one to conclude on. It's easier when you're comparing segments from the same individual. Then we'll say this ileum has much more Peyer's patches compared to this jejunum that has fewer of the same individual. It will be more objective if they are of the same individual at the same magnification, then we count. If it's just single like that, I agree with you, it's too abstract. But the other concepts you can still apply and it will be okay. The fact that we have the villi tells you the small bowel. Then if you have a longitudinal section, the fact that the plica circularis are widely spaced, that one makes sense. If we want you to pick a segment of the small intestines, and specifically ileum, we will then intentionally pick a region in circular region that has several lymphatic nodules. And in that, you'll be able to see even more than 4 lymphatic nodules in the same view. And then you know that this is actually several. Okay?
Okay.
All right. Okay, Victor, you have a question?
Yes, regarding the villi and the plica circularis, are the villi mucosal foldings, and are the plica circularis foldings of the submucosa?
Yes. So villi are folds of the lamina propria. Plica circularis are folds of the submucosa.
Okay.
But all of them, the mucosa will still have an impression of a fold. You see, if the submucosa is folded, even the mucosa will have the fold grossly. If the lamina propria is folded, even the epithelium of the mucosa will still have the fold. But villi are thickening and projections of the lamina propria. And then plica circularis are projections of the submucosa.
Thank you.
Welcome. All right, you can proceed now. We are now going to look at the endocrine secretions of the stomach and the small intestine. I want to bring this up because we've now talked about histology of the stomach and histology of the small intestine. And I've mentioned to you that we have some cells which produce hormones belonging to the enteroendocrine cells. I think it's important to just consolidate that story in 1 or 2 slides. So take note that there are several hormonal substances in form of chemical messengers, or there are several chemical messengers in form of hormonal substances which are released into the alimentary canal, or rather into the bloodstream around the digestive system. These hormonal substances are produced either by the nerve terminals within the gut wall, which means the enteric nervous system, or they're released by epithelial cells within the gut wall. The epithelial cells in the gut wall that produce these hormones are the ones we call the enteroendocrine cells. The regions where we have much hormones would be from stomach, duodenum, and the jejunum. There are several cell types which have been identified in the gut mucosa responsible for producing hormonal substances. These chemicals will pretend to work in a paracrine manner. And when you say paracrine, it means that the secretions are released into the extracellular fluid, and these secretions move through diffusion within the extracellular fluid to affect neighboring cells. So that is paracrine. However, some of these chemicals enter the bloodstream, and which means that they can be transported far places from the site of production, and that makes them hormonal. They form part of what we call the diffuse neuroendocrine system of the body. The diffuse neuroendocrine system refers to a system where we have cells which are scattered, but they produce hormones into the bloodstream. These cells are stimulated by the nervous system. So that's why we call it the neuroendocrine system. Previously, it was known that these cells use amines as their precursors, and they cause decarboxylation on these amines. And so these cells were known to be what we call the amine precursor uptake and decarboxylation system. They're falling in that system of amine precursor uptake and decarboxylation system, abbreviated as APUD system, APUD system. We don't quite use that term so much currently because we know that not all of them may necessarily just take amines and decarboxylate the amines, but they are part of the diffuse neuroendocrine system. If you are to bring all the endocrine cells of the alimentary canal in one block, they could probably constitute the largest endocrine organ in the body, but they are not in one particular aggregate. They're diffuse. So what are some of the hormones which are produced by the gut? We have gastrin. Maybe I forgot to tell you that most of these hormones influence the functions of the digestive system, and the physiology team will do more justice than what I've done in that particular statement. Gastrin is one of the hormones which is produced by the G cells. The G cells are found in the stomach, G for gastric, and in the duodenal wall. The role of gastrin is to stimulate gastric acid production. It also stimulates the production of pepsin, the enzyme. The other function does is stimulate the mucosal growth of stomach and intestine, so helping the stomach and intestine to regenerate its epithelium. So basically helping protection. We have cholecystokinin, commonly abbreviated as CCK. Cholecystokinin comes from the eye cells, which are found in the duodenum and jejunum. The eye cells producing cholecystokinin are largely present in those two regions. They're stimulated by a number of things. For example, the presence of fat within the alimentary canal and the like. After producing CCK, CCK is known to perform a number of functions. Its name being given to it because of its role in causing contraction of the gallbladder. Well, that's not the only function. The other functions we know is that it stimulates production of pancreatic juice, particularly the enzymes and the bicarbonate from the pancreas, so pancreatic juice. It also stimulates relaxation of the sphincter of Oddi. Remember that the sphincter of Oddi is the one that releases or control release of substances from the main pancreatic duct as well as common bile duct into the D2, second part of the duodenum. So it makes sense for this hormone to cause gallbladder contraction as well as relaxing the sphincter. So this is helping that function to be harmonious. Secretin from the S cells of the duodenum and the jejunum increases the production of bicarbonate-rich content of the pancreatic and bile juice. So largely the bicarbonate-rich component of those pancreatic and biliary secretions. It also decreases gastric acid production. Gastric inhibitory peptide is from the K cells with the duodenum and the jejunum. They stimulate insulin secretion. Well, that's their major part. So we consider that as a pathway independent, a pathway of stimulating insulin that, generally speaking, is not directly going to the beta cells. The glucose concentration directly is not directly affecting the beta cells. It is through another channel. Motilin from the MO cells of the stomach, small intestines, and we're adding colon here. Motilin, as the name suggests, promote contractions, motility. And so it regulates what we call the migrating motor complexes. These are just contractions of the alimentary canal. You learn more from the physiology team. And so this helps to control motility between meals. Somatostatin from the D cells. Remember, D cells are present both in the pancreas as well as in the alimentary canal and largely stomach, small intestine. Somatostatin inhibits acid production. It inhibits gastrin production. It inhibits gastric inhibitory peptide. It inhibits secretin, motilin. It inhibits pancreatic secretion. It also inhibits some functions like gastric motility. It inhibits gallbladder contraction. It inhibits food absorption. So it's an inhibitory hormone for multiple functions. somatostatin. Statin for static, stop. Somato for body, so it may bring some functions to a stop. Okay, that summarizes the story of stomach and small intestine. Now let's go to large intestine. I'm projecting for you a slide of the colon. Somebody to tell me one reason why this is colon. Yes, again.
Oh, presence of this is the three, okay, the tinea coli, the three bands of longitudinal muscles where you can see. Yeah,
Very good, very well. So we can see here we have a thickening. We have another thickening there, and we have another thickening there. We can see tinea coli very well. So tinea coli are thickenings of the longitudinal muscle, and there are three. So we can see that one there. So this is Muscularis externa, then this is submucosa, then this is mucosa. Now, what is unique about the colonic mucosa? Okay, let me give this to anyone. What is unique about this colonic mucosa? Yes, Michelle.
We can see the amount of goblet cells is a lot, and we know that increases the lower you get in the alimentary canal. Are these also crypts of Leeuwenhoek?
Yes, so we can see several goblet cells. I agree with you on that. And we can see the intestinal crypts of Leeuwenhoek as well. The difference here is that colon does not have the villi. And so the crypts of Leeuwenhoek of the colon just open onto the surface, not necessarily adjacent to villi. So that's a crypt. That's a crypt. That's a crypt. So these are crypts. And essentially, if you just follow the outline of the goblet cells, those are basically the crypts. Okay. Is that okay, Michelle?
Yes, yes.
Okay. Yes, so that is basically how colon looks like. You pick it by the fact that you have the tinea coli and you have the crypts lined by several goblet cells. Okay, another segment. Anyone to give me a trial on this one? I have two magnifications of this. This is the low magnification, and that's the higher magnification of the mucosa. So you're basing your judgment on that as well as that. It's one organ with different magnification. Anyone wants to try? Yes, again?
Okay, so then I'll go back to like what Weru taught us, not taught us because we knew that, but then based on when you opened it to a higher magnification, I saw the binucleated. So I'm thinking it's a transitional epithelium. Yeah, right here. Okay, you can't see me. But like for my head.
Very well, very well. So this epithelium has transition epithelium still. We can see binucleate cells there. So definitely not alimentary canal. Yes, this is still ureter. It's not alimentary canal. So the learning point, I don't want you to pick that's ureter. The learning point that I want to pick that this is not alimentary canal. All right, another segment. Anyone? So this one has 3 magnifications. Let me take you through the magnifications, then you tell me what you think. So this is the wall. and this is the lumen. There's some things to note at this magnification. One is that the lumen has some material within it. That's important. Two, the mucosa has several lymphatic nodules. We can count, let's start from 12 o'clock. So maybe that's one. I'm not fully convinced about that. We can skip it. Maybe it is, maybe not. But definitely that's another one. 3, 4, 5, 6. Not convinced about that and that as well. Even here, maybe that's another one, not so sure. But at least we can see several lymphatic nodules. That's the point. That is this magnification. Going to the second magnification, we confirm that yes, we have the lymphatic nodules. Yes, we have some material within the lumen. From there to there is the submucosa. Then this is Muscularis externa. We can see inner circular orientation and outer longitudinal orientation. And we can confirm that the structure is intraperitoneal because you can see the serosa there. Yes, Mark.
So I'd want to guess, I think this is the vermiform appendix.
Okay.
Because we have the lymphatic nodules, but we lack villi.
Very well. Thank you. Yes, this is the vermiform appendix. And the third magnification is that of the epithelium. So we can still see the intestinal crypts. We don't have much of the goblet cells though, but yes, we can see the intestinal crypts there. We don't have the villi. We can still see the material within. So the learning point is that appendix usually has some material within it. Could be impacted food, could be impacted mucus, but usually it has some material within its lumen. That's a bit redundant all the time. That also applies to rectum. You may also find some material within the lumen. Now, the one for the rectum is not necessarily stool, could just be a lot of mucus in the rectum. All right, so this is vermiform appendix. Try another one. So 3 levels of magnification for this one as well. In this magnification, this is the lumen. We can see this is the mucosa. And we can confirm that the mucosa has some folds which have been created by the submucosal extensions. So this is a fold, that's a fold, that's a fold, that's a fold. This being a cross-section, and we see those folds. How would you describe these folds if you were to describe them in 3D? Yes, Como.
I would describe them to be longitudinal, running parallel to the length of that particular structure.
Very well. So we are seeing a segment of the JT that has longitudinal folds. Como, what do you think? What part of the JT could this be?
If it has longitudinal folds, we have 2 choices. It can either be the rectum or the esophagus.
Okay, and to distinguish the two, I give you high magnification. so that you can see the mucosa. So what do you make note of that?
I would say that the mucosa is lined by the crypts of labor coon, and it contains very thick secretions of mucus. Therefore, I would definitely say this is rectum.
Yes, so the epithelial lining here has several goblet cells. We don't expect that in the esophagus. Yes, we can see that, and this is a submucosa. Again, this submucosa does not have glands. It's not esophagus. This is the inner circular layer. That's the outer longitudinal layer. Perhaps this region shows the mucosa better. In the lumen, we can see very thick mucus secretion. So this is rectum. This is another image, not of the same slide, but just to show you how goblet cells can be numerous. These are goblet cells, and this is mucus plug. That is very characteristic of the rectum. Great. Now I want us to finish the story by identifying some slides. So I'll show you one slide. You tell me which part of the JT that could be. Let's start with this one. Just tell us, what do you think and why? Yes, Michelle?
So these projections into the lumen that I'm assuming are villi. So because of that, you can see that this is possibly the small intestine.
Very true. Do you want to be more specific than that?
Specific.
Yeah, you said small intestine. Yeah. Is it duodenum, jejunum,
I'm
or
not too sure, but in the submucosa, I think this might be the duodenum because of the presence of, I'm not sure if I'm seeing glands in the submucosa. Yeah, that's my best.
Okay, actually there are glands, Michelle, you're right. So these are submucosal glands. That very thick zone, those are submucosal glands, the whole of that. So that is duodenum, you're right. Okay, another person. So this is just the mucosa of a segment of the JT. Which segment of the JT could this be and why? Yes, Winnie?
I'll say that it's a colon mucosa, since they are very long crypts of liver cone that are lined by numerous goblet cells, and the crypts of liver cone are not opening, not exactly to the villi, but to the surface. Yeah,
okay, very well. So we can see intestinal crypts of liver cone lined by goblet cells, several goblet cells, and there is absence of villi. This is definitely large bowel. So it can be colon, could be still rectum, but more classical for colon because of the crypts of liver cone. Okay, next one. Yes, again.
So presence of villi, so we know it's a segment of the small intestine. There are no submucosa glands, so then it's not duodenum. So it's either jejunum or ileum.
Okay, and I agree with you on that. It is either jejunum or ileum. With this orientation, difficult to distinguish whether jejunum or ileum. So It's either jejunum or ileum are green. Okay, so this is a longitudinal profile of a segment of the JT. Anyone to try? Okay, I'll give it to Anton.
This is the ileum.
Okay.
Yeah.
Reason?
There's villi present, and then the space between the rings. Okay, I can't see the other ring, but there's so much space left.
Very well. So very true. The fact that you can see villi and then you can see there is a fold and you don't see the next fold tells you that the next fold is a bit far.
Yeah.
And of course, we don't have submucosal glands. So this is ileum. Okay, that's the next one. Anyone to try? Okay, okay, at least. Tamara, let me give that to you.
Okay, it's just a trial, but I think that this is the stomach. I can see like gastric pits almost, yeah. And it also has like a relatively thick muscular wall. And the ducts, they're like ducts opening into the gastric pits.
Very well, you're right. So these are gastric pits, several of them, with several mucosal glands, and we have very thick muscular layer. So yes, this is a gastric wall. Anyone for this? I'll give it to Winnie.
I'll say it's the vermiform appendix, majorly because I can see numerous lymphoid aggregations. There are no villi, and there's some material inside the lumen.
Very well. So yes, we can see lymphoid nodules, several. There's material in the lumen, and there are no villi. This is most likely the vermiform appendix. Okay, this one. Yes, Sydney.
I'll say it's the colon.
The reason?
Because I can see the bands of tinea coli, the 3 bands.
Very well. That's the most outstanding thing at this magnification. So we can see 3 bands of longitudinal muscles. This is a colon. Victor, you have a question?
Yes. Could we say that it's the colon also because of a wide lumen, in addition to the tinea coli?
Yes, good observation. We expect the colon to have a wide lumen. So if you look at the size of the lumen vis-a-vis the thickness of the wall, that is okay. That is still a point to note. I would, however, be uncomfortable to just give us wide lumen as the only reason. It may not be a standalone. In the context of tinea coli, it confirms it. Yes,
How do you identify the colon from the cecum?
so basically, the cecum will have even much wider lumen, but cecum doesn't have the tinea coli.
Okay, thank you.
Okay, we have another one here. Yes, again.
I'm going to say the vermiform appendix because I can see lymphatic nodules.
Okay, still vermiform appendix, yes. Multiple lymphatic nodules, then a material, and then the villi. Anyone for this? Yes, Michelle?
I would say that this is definitely the large intestine because there is deposits of secretions of mucus at the top. We can see the crypts of Liverpool lined by the goblet cells and these villi.
Very well. Well stated. Crypts of Liverpool lined by goblet cells, then no villi. This is large bowel mucosa. Great. So that is it for that session. We will have another session similar where we look at histology of the extrinsic glands of the digestive system another time, most likely next week. So I'll stop there and give you opportunity to ask questions if you do have.